This is Win Arias. I'm a senior scientist emeritus at the National Institutes of Health, and it's a pleasure to invite you to the fifth session of the 25th year of a novel course called Demystifying Medicine, which is conducted from the NIH in Bethesda, Maryland, and involves participants from around the world. The purpose of this course is to bridge the exciting developments in biology, engineering, and computer science with medicine. It shouldn't matter what field of research or interest you specifically are involved in. The topics that are presented and the advances that accompany them should be of interest to people working in any area of biomedical research. So the intent is to create a bridge, as illustrated in the slide, the Brooklyn Bridge. And in fact, we every week are like the folks on the catwalk, where individuals representing two very different approaches to major biological medical problems are sharing ideas. Once a bridge has been completed, life is never the same on either side. And we hope that that's the case when people collaborate and learn about areas that are on the surface seemingly very different from the major areas of their daily focus. The course is held from 3 to 5 p.m. Eastern Standard Time from January to May. And you are watching it on the NIH video cast. For those looking for CME credit, it is available, and the code for today is 67270. Now, we encourage people here at NIH and all over the world who are watching this program to submit questions during the presentations. And you do that by selecting the send line feedback on your video cast display. These questions will be grouped together and presented to the speakers at the conclusion of their talks and form the basis of a continued and invariably lively and highly productive discussion. I bring to your attention that all previous sessions back to the very beginning are archived, and the link is presented on this slide. If you request additional information, we give you our email address. Next slide, please. Now, today's topic may seem somewhat esoteric to some, but it has a much bigger format. It raises the interesting question of whether cells, particularly epithelial cells, which in an organ all look pretty much the same, but do they do different things? And if they do, what controls their individual function? Now, a few facts about hepatic physiology that are critical. First of all, the liver is primarily a hematologic organ, beginning from a hematologic basis and then invasion by what becomes hepatic cells. And it receives about 2 liters per minute of cardiac output, and that's about 25% of the entire cardiac output. The liver itself is generally about 10% of the body weight, and it's the largest single organ. A few other facts. It has an interesting blood supply. Arterial blood mixes with venous blood entering into the liver, and then eventually passing back to the heart. And in this process, it bathes the hepatocytes, which form, as you will see, a linear arrangement called a sinusoid. Those cells all look alike. And interestingly enough, you never or rarely ever see them divide in any normal liver tissue. What's the lifespan of these cells? And why, when in the face of hepatic damage or resection, the entire lobule begins a wave of replication? Another point, these cells are only they're the dominant of the 6 different cell types in the liver. Now, how is this structure maintained, regulated, replaced? And the concept of zoning came about actually almost 100 years ago when Aaron Rappaport in Toronto, who studied the blood supply, proposed that there were different areas of oxygenation. Little was done until the period of the 1960s, when Alex Novikov and Edward Essner and others applied enzyme histochemistry, particularly for various phosphatases and phosphorylases, and described entirely different populations within these putative zones. Next, it was noted that with certain diseases, and in particular toxicities, only specific areas, zones, were affected. And attempts were made to use that to separate cells from different zones. By and large, these experiments were more difficult than anticipated and did not yield very rewarding results. The real breakthrough occurred with the development of the 2 technologies that are going to be discussed today. The first is the development of infravital and supravital microscopy using fluorescently labeled various gene products to actually determine their distribution, function, and so forth. And that is going to be discussed by our first speaker. The second major advance, of course, is the ability to conduct genomic sequences in individual cells. And this gets closer to the central issue, the big biological issues. Where do these cells come from? How do they replicate? Why do they respond as they do to injury? And what is the mechanism for zonation? How is it regulated? And as we'll hear today, how it's related to various disease processes, at least conceptually, such as cancer, cirrhosis, and so forth. So we're very fortunate to have 2 speakers who are clearly at the forefront of this. Now, the first is Natalie Porach-Leon. Natalie received her degrees, undergraduate and master's degree in Israel at Tel Aviv University, and a PhD in cell biology. She was part of an NIH graduate partnership program which brought her here, and she worked with Julie Donaldson in the National Heart, Lung, and Blood Institute, and then had postdoctoral training with Roberto Weigert in the National Cancer Institute, where she became a master of intravital microscopy. She is recipient of an NIH Pathway to Independence Award, and currently is a tenure-track Statman investigator in the National Cancer Institute. Her research employs a very integrative approach involving molecular biology, biochemistry, and cutting-edge imaging methods to examine the fundamental principles of zonation and their consequences. She's very active in various groups and activities here at the NIH and has been widely sought after as an invited speaker on hepatic zonation at major medical meetings and institutions. Now, our second speaker is her colleague, Shela Vitzkovich, who also received his training in Israel. His bachelor's degree was in physics and mathematics at Hebrew University, a master's in electrical engineering at the Technion, and a PhD in biology with honors from the Weizmann Institute of Science in Rehovot, Israel, where he works at present. He did postdoctoral fellowships at the Weizmann and also at MIT. He's the head of the Department of Molecular and Cell Biology at the Weizmann Institute, and his research involves applying tools from systems biology to study the design principles of mammalian tissues, including hepatic zonation. He seeks to understand how the structure of tissues and their single cell gene expression patterns serve to achieve physiologic goals, and how these interactions are disturbed in disease.
Is Dr. Arias frozen, or perhaps I am frozen?
Arias.
I see. Well, almost done. Perhaps I'll stop sharing. Is Dr. Arias frozen, or perhaps I am frozen? Okay, I'm going to stop sharing, and Natalie, maybe you can begin.
Sounds good.
That's coming up nicely.
Okay. Can you hear me okay?
I can hear you, yes.
Okay, so thank you very much for the introduction and the invitation to present our work. The title of my talk today is How New Tools Made Liver Zonation Visible and Why It Matters for Cancer Research. So today's story is really a story about how an old histology observation became a modern spatial biology framework. And during my talk today, I'm going to have 3 sections. I'm going to introduce and define what is zonation. I'm going to tell you about how technology gradually revealed zonation and made it more visible and accessible. I'm going to tell you how my lab is working hard to move away from transcriptional maps and pinpoint functions. And at the end, I'm going to wrap it up with talking a little bit about why it matters for liver disease, for cancer, and beyond. So let's get started. Before we can talk about zonation, I would like to introduce some concepts that relates to liver anatomy, and Dr. Arias did that a little bit. Hepatocytes that are the major cells in the liver are organized in these hexagonal units shown here, and I'll refer to them as the liver lobule. And within each lobule, blood flows in one direction. The blood comes in through the corners of the hexagons and flows through the corners towards a single central vein. And we can take a closer look at this portal to central axis, meaning the areas where blood enters the liver lobule is called the portal area or periportal. The liver is fed with 2 blood supplies. One is from the hepatic artery, which is rich in oxygen, and the other one from the portal vein that is rich in nutrients. The blood flows directionally towards a single central veins. And as you can see here, the hepatocytes are essentially sitting next to each other along these blood vessels. Each axis is approximately 200 to 300 microns apart. And one question that could come to mind is the hepatocytes that are sitting along this axis, are their functions and behaviors similar or different? And the initial observations that they might be different came as early as the beginning of the previous century, where scientists noticed that the cell biology components of the different cells in the liver looks different. And what I'm showing you here is a hand-drawn figure. Back in the days, they used to do that. And the scientist is describing essentially the glycogen content within the hepatocyte's cord, moving from the portal where the blood enters towards the pericentral in different organisms: rabbit, guinea pig, mouse, chicken, etc. And what they noticed, and you can see here in the schematics, is the glycogen content was variable. Another thing that was visible to scientists and cell biologists using microscopy is that the mitochondria morphologies were very different in different parts of the liver lobules. And I'm showing you here a close-up of a periportal mitochondria that is marked with a dashed red line, and it's small and round compared to the pericentral mitochondria that were more tubulated and long. So these early observations from close to 100 years ago led scientists to coin the term liver zonation, which is essentially the spatial pattern of gene, protein, and metabolites expression and function along the portal to central axis. But the reality is that this idea was very difficult to measure. And for years, liver zonation remained a concept that pathologists used mainly to describe where a lesion or a disease was located, whether it was in zone 1, closer to the portal side, or zone 3, closer to the central side. But these were arbitrary divisions. And if you look at a tissue, a liver section, and you zoom in on this axis, there aren't clear boundaries. So in the tissue, you don't have physical division of the three zones, which essentially meant that it's impossible to isolate them and apply traditional biochemical techniques. And therefore, liver zonation remained in the realm of physiological phenomenology that wasn't really integrated into liver physiology and biology research, because scientists kept on grinding whole livers and averaging everything out. And the history of liver zonation research is really mirrored by technological progress that drove the discoveries. And this is a summary of some of the main techniques and where they appear in the timeline, on the timeline, from the initial EM electron microscopy observations around the 1950s, going through an era where dissecting microscopes and immunohistochemistry allowed to look at create essentially maps of individual enzymes or measure an individual enzyme activity in space. But the information that we were able to gain from these kind of techniques was very, very limited. And as Moon mentioned in his introduction, the real revolution came less than 10 years ago when Shalev and his group dissociated hepatocytes from the liver, did single-cell RNA sequencing, and based on landmark genes, essentially reassembled the liver lobule back. And I'm sure he's going to tell you way more about this seminal study. But what we learned from the spatial, the single-cell study is that now we essentially have the gene expression map of every hepatocytes along the portal central axis. We know that half of hepatocytes genes are not uniformly expressed, which is pretty striking. So 50% of the genes are different between the different hepatocytes that are sitting very close to one another. We know that the spatial separation, or these expression maps, are telling a story where certain genes that engage in certain metabolic processes are separated in space. So incompatible chemistries or metabolic or conflicting metabolic processes, like, for example, the synthesis of glucose or the usage of glucose, are separated in space, as shown down here. So you have this division of labor, and that explains how the liver can do so many things. And another interesting thing that Shalev discovered and single-cell technology and transcription sequencing taught us is that zonation is not a static state. It's dynamic, it's responding to feeding and fasting, which explains why you don't want to have membranes to restrict the zones. But many of these functional conclusions or metabolic pathways that we write down here are still inferred based on of expression, and they're not directly measured. And we all know that RNA is really the blueprint, but if a gene is expressed, it doesn't mean that the protein is there, and it doesn't mean that the protein is actually working, because there's another layer of post-translational modifications. And that is really where my lab comes into play, where our goal is to move away from or use the transcriptional maps and combine them to really get to the function. Because the general theme in my lab is when you understand function, you can understand what goes wrong in disease and get better insights into the mechanisms. So the gaps are how the transcriptional maps that we have now in great details are becoming physiology in vivo. And the overall hypothesis is that post-translational regulation, organelle architecture, and organelle interactions can convert these gene programs into metabolic outputs. And I just want to make the case that the organelles, the cellular organelles, are the factories and is the site where metabolism takes place. And then the lead in is the mitochondria. It is the ideal one to study because many of the zonated liver functions are centered in the mitochondria. And as you remember, a few slides ago, I showed you that these observations of the mitochondria were one of the things that scientists noticed that made them coin this term of liver zonation. But can we dig deeper, understand what are the functional consequences of this structural variabilities, and also try to understand what creates them and how they're maintained with time. So luckily, the tools, and in this case, our ability to perform electron microscopy has significantly improved. And today we're able to perform not only high resolution electron microscopy images, but actually take these images in 3D. And what you're seeing here is essentially a block of a periportal cell versus a pericentral cell. And what I hope you can see here, aside the fact that the data set is beautiful, is that the mitochondria are fundamentally different. And aside for the difference in the sphericity, namely that the periportal ones are round and large and the pericentral ones are tubulated, what I hope you can see, and this is totally lost in the 2D images from 100 years ago, are these structures that look like folded red blood cells that I'm going to come back to later on when I'm going to talk about organelle contact sites. So aside for the morphology and the spatial organization, what else can we do? What additional technologies can we develop to learn and get closer to the function? And what we did here is was inspired by another technique that was developed in Shalev's lab, taking advantage of the fact that different hepatocytes have different surface markers expressed, as shown here. You can see that the E-cadherin in magenta is primarily localized to the periportal regions, while the CD73 is enriched in pericentral hepatocytes. What we did is we isolated the primary hepatocytes from a mouse. We labeled them using these markers in a tube, and later we fax sorted them, again, based on the expression, to generate essentially two groups of enriched populations, either periportal or pericentral. And what I'm showing you here is a Western blot showing you the expression of different markers, either periportal or pericentral, demonstrating that this approach is very effective in enriching the different populations of cells. Now, we've done several things to, several studies with these cells to characterize their mitochondrial function. But what I want to focus on today is how zonation is regulated. And one of the things we did with these two populations of cells was to perform proteomics and phosphoproteomics analysis. And what I'm showing you here are all the phosphoproteins that we identified in the two populations, presented as a ratio. And what I hope you can see, that there are certain phosphosites on proteins that are enriched in pericentral hepatocytes, and there's a different set on periportal side, which means that post-translational modification, phosphorylation, is also zonated. Now, we used different bioinformatic tools to look deeper and characterize what these phosphosites were related to, which pathways, which processes. And the two proteins that came up the most were AMPK and mTOR and their downstream effectors, which made us wonder whether nutrient sensing signaling, so AMPK and mTOR are two very central nutrient sensing kinases in cells, and their activation is regulated through nutrient availability or the lack of nutrients. And they activate a cascade of phosphorylation. And in order to examine whether nutrient sensing signaling pathways play a role, we decided to activate AMPK and mTOR, and this is what I'm going to show you now, acutely, in vivo, and look at the morphology of the mitochondria. So when you inject the vehicle of these drugs, you see the periportal, like I showed you before, are round and spherical. The pericentral is more tubulated. If you activate AMPK or mTOR, you essentially able to switch or shift the mitochondrial morphologies. With AMPK activation, the mitochondria become round and spherical. With mTOR activation, you see the elongation, and I hope you can appreciate how much more dense the mitochondria content is here. And this is essentially a quantification demonstrating how round or elongated a structure is. So what we concluded from these studies is that not only we could see that the mitochondria from different parts of the lobule are morphologically different. We could measure that their activity was different, but we were also able to manipulate their morphology with activation and inhibition of nutrient sensing signaling, which indicates that perhaps along the periportal pericentral axis, there is a nutrient gradient that helps maintain mitochondrial zonation, mitochondrial heterogeneity. And again, this concept of a gradient as opposed to a membrane, a physical separation, as a way to regulate the heterogeneity in the liver is a new concept. So we next wanted to look at what happens when we change the diet. Okay, so what happens if we feed, we have a way to manipulate the diet in mice. We can feed them Western diet to increase the lipid flux. Feeding with control diet, which is what I'm showing you here, you can see that the liver is not a lipid storage organ, and there are very few lipid droplets in the tissue, and many of them are concentrated around the pericentral regions, which is where we think lipid synthesis takes place. If we feed the mice a Western diet, which is high in lipid, in cholesterol, and in sugar, what you see here is an accumulation of lipid droplets in the tissue. This is the equivalent of simple steatosis in patients, or what is known as MASLD, or formerly known as fatty liver disease. What is less known is that if you take mice and you fast them overnight, you can also increase the lipid flux into the liver. And that is because nutrients are unavailable and adipose tissue is undergoing lipolysis, and those lipids are transferred into the liver to sustain whole body metabolism. And if we look at the livers of fasted mice, I hope you can see here that there is a significant amount of lipids accumulating in the liver. So under these three different nutritional conditions and three levels and sources of lipids in the liver, we ask the question, where are the mitochondria? And what happens to the mitochondria zonation? And so what we found is that Western diet induces rounding and fragmentation of the mitochondria. They are very rarely in contact with lipid droplets, as shown here in this cartoon. On the other hand, in the fasting conditions, mitochondria are elongated, and they're often seen wrapped around lipid droplets, as shown here and down here, and in this cartoon, which one made us visualize that the mitochondria heterogeneity along the liver lobule was lost. Western diet or fasting, either increasing the lipid flux into the liver, caused the mitochondria to lose their zonation heterogeneity and gain this one specific phenotype. But it also led us to ask the question, what is the role of these contact sites between lipid droplets and mitochondria, and whether these interactions are beneficial in lipid management? So when do these interactions come into play? Lipid droplet and mitochondria were shown in the literature to facilitate lipid storage. Mitochondria provide the energy that is required for triglyceride synthesis. In other model systems, these interactions were shown to facilitate lipid utilization. Fatty acids are released from the lipid droplets and are oxidized in the mitochondria. And this close proximity essentially allows for the fatty acid transfer in either direction. But what is happening in the liver was not entirely clear. In our proteomics study, we discovered upregulation of perilipin 5 in the context of fasting, where we see enhanced contact sites between lipid droplet and mitochondria. And this perilipin 5 has two domains that you need to know of. The first one is the phosphocyte. It was shown to be phosphorylated by pKa. And another one is this mitochondria recruitment domain that was shown to, again, allows the perilipin 5 that is associated with the lipid droplet to recruit mitochondria and form these contact sites. We were very fortunate to be able to receive perilipin 5 variants from Sarah Cohen at UNC, and we decided to try to manipulate these contact sites in vivo. What we did essentially was to express a full-length wild-type perilipin 5, a phosphonull, a perilipin 5 that cannot undergo phosphorylation, a phosphomimetic perilipin 5, and a truncated version of perilipin 5 that is unable to recruit mitochondria to lipid droplets. So essentially, we took these constructs, we injected them into mice, either fed controlled diet or Western diet, and the goal was to remodel mitochondrial lipid droplet interactions, to observe mitochondria morphologies, and ask the question of the role of mitochondrial lipid droplet contacts in vivo. And so what I'm showing you here is a representative image of a periportal, midlobular, and pericentral hepatocytes. Mitochondria are in green, lipid droplets are in magenta, and this is the analysis of the interaction or the overlapping pixels of the different constructs across space. And the main conclusion here is that the perilipin 5, the phosphorylation site of perilipin 5 is critical in being able to form the mitochondrial lipid droplet contacts, as you see here. Unphosphorable perilipin 5 that is unable to undergo phosphorylation is intimately in touch with the lipid droplets, and I hope you can see that they're way larger than all of the other conditions, while the E mutant is these contacts are detached and the lipid droplets are rarely there, suggesting that the contact sites are promoting lipid droplet storage throughout the lobule, and the E is promoting lipid droplet usage, or maybe even fatty acid oxidation. So we measured triglyceride content, and indeed in the A mutant, we can see higher triglycerides content in the tissue. And then we asked if these contact interactions can counteract some of the damaging effects of Western diet. And to make a long story short, the answer is yes in the short term. So if we express the E mutant, oxidative markers increase, while when you express the A and you form these contact sites, lipid peroxidation and damage is reduced. But we also found that long-term Western diet feeding is associated with enhanced mitochondrial lipid droplet contact sites. So with chronic exposure, these contacts that do promote lipid storage and expansion also promote liver disease. So to summarize this story that I presented really quickly, we think that under homeostasis, zonation is visible. Periportal mitochondria are spherical and round. They're oxidizing fatty acids. The pericentral ones are more tubulated, and if you remember in the 3D, they had these little cups. Those cups are the contact sites with the lipid droplets that allows minimal expansion and content of lipid droplets in pericentral regions under homeostasis. If you constitutively phosphorylate perilipin 5, the contact sites are detached. This is mimicking Western diet feeding in the short term, where the mitochondria are trying to consume the fat, the excess fat, and oxidize it. But in the long run, this is, but essentially this over-oxidation of fatty acid can produce damage. And in the short term, if we force the lipid droplets to come in contact with the mitochondria, the excess lipids can be stored in lipid droplets and have some protective element to the tissue. And so this is a good segue. I talked about Western diet. I talked about liver damage and trying to protect the liver from lipid damage. Why do we need to study zonation in the context of liver disease and cancer? And is it important? And so if you look at liver pathology, and I summarized several liver diseases down here below, you'll notice that the disease onset and the pathology patterning is also zonated. I just showed you MASLD and lipid droplet accumulation, but what about the interdependence between zonation and liver cancer, and which is a question that we're extremely interested in here in the lab. So I want to wrap up my talk by highlighting a recent study that used a very sophisticated mouse genetic approach to ask the question whether all hepatocytes are equally likely to give rise to HCC, to hepatocellular carcinoma. And Kate Burrows from my lab and myself just wrote in the Spotlight summarizing this beautiful study. And what the Zhao group at UT Southwestern did is to use these Cre-Lox mice to create essentially zonated liver cancer. And this is shown here in this cartoon. The pre-malignant lesions are shown in green here, and the red mask to the cells is showing the restricted areas in which these tumors were created. So they compared essentially cellular transformation in periportal regions versus pericentral regions. And what they found was very interesting and surprising. They found that periportal tumors grow and expand very rapidly, but they rarely progress to HCC, essentially to hepatocellular cancer. While the pericentral ones, even though the clones were smaller, those were the ones giving rise to HCC. And mechanistically, they explain this by this zone 3 expressing GSTM 2 and 3 enzymes. These are enzymes that are involved in detoxification, essentially removal of lipid peroxidation and other damaging agents. And when the GSTM 2 and 3 are active, they essentially reduce ferroptosis, which is a form of cell death, and protect the cells. Now, pericentral cells or zone 3 cells are more susceptible to experiencing lipid peroxidation and oxidative stress. which is why GSTM2 and 3 are more active and highly expressed there. But in the context of liver cancer, this protective physiology is actually what allows tumors to survive and progress to HCC. And this study did a very nice job in inhibiting and deleting these enzymes and showing essentially that liver cancer is a zonated disease and should be considered as such. So I described how new technologies changed the field from classical microscopy to single-cell spatial methods and advanced imaging that led us to connect maps of genes to functions. I provided examples of how zonation works without physical boundaries. I mentioned that we think that the nutrient gradients along the axis serves a role in regulating zonation. I showed you post-translational modifications. I showed you organelle interactions and architectures. And we think that studying all of these cellular processes in the context of zonation is fundamentally important to understand liver health and disease. I also gave you at the end this opportunity to apply zonation lens to liver disease and cancer. I think this is a good opportunity to finish with the question, does this similar built-in geography or zonation matters in other organs, and should we really shift from studying organs and cell functions within organs by averaging all the cells and grinding tissue to using more spatially oriented technologies? With that, I want to thank my lab who did the work, our collaborators, and the NCI course. Thank you.
Thank you. And I welcome Dr. Arias to join, if possible, for the transition. I'm not quite sure if he's set up yet. Okay, if not, we can just ask our next speaker to come on and start sharing his slides. They're coming up. Excellent.
Can you see my slides?
I can see. Can you speak again? It might be a little low.
Yeah, can you see my slides and hear me okay?
I can see your slides clearly. You're a little low, but keep talking. You're good.
Okay. I will try to speak loudly. Okay, so thank you very much. Yeah, thank you, Win, for the kind invitation, and thank you, Natalie and Win, for the introduction on liver zonation. I'll tell you about some of the work we've been doing in the lab and trying to map liver zonation across space in mouse and human. This is a disclosure. I'm the founder of Trace Cells. The liver is the most diverse organ in our body. It's a multitasker. It has about 500 different functions. For example, it stores nutrients after we eat and releases them in a regulated manner in between meals. It detoxifies blood, produces all the carrier proteins in our blood, clotting factors, complement system proteins. It produces hormones. And all of these functions are performed by the same cell type, the hepatocyte. I want to highlight one very important function of the liver. It's being called the glucostat of our body. In our blood, at every minute of the day, we have about 4 grams of glucose, whereas in a single meal, we digest the equivalent of something like 30 grams or 40 or 50 grams of glucose. These are many carbohydrates that are broken up in the gut into glucose, and our blood is flooded with dozens of grams of glucose, but the liver very effectively absorbs all these glucose molecules, integrates them into glycogen, and makes sure that we remain at 4 grams of glucose. And when we jog, when we feel bad about all the carbs we ate, our muscles take up about 30 grams of glucose from the blood for the excess energy, but blood glucose levels will remain at about 4 grams. And again, this is because the liver is the glucostat. It knows how to release just the right amount to maintain glucose homeostasis. So this is one type of homeostasis, but the liver is basically responsible for homeostasis of many other physiological features. So 500 different functions, all of them performed by the hepatocyte, but as we've heard, the hepatocytes are extremely diverse. They live in these lobules that are graded microenvironments that have gradients of oxygen, nutrients, hormones, morphogens. And if you stay in the liver, as we've done here with a technique called single molecule fluorescence in situ hybridization, this is a technique that reveals individual RNA molecules as diffraction emitted spots. You can immediately appreciate that periportal hepatocytes around the portal vein are very different than pericentral hepatocytes. You can see that the periportal hepatocytes express this green gene, CYP2F2, and the pericentral hepatocytes express this red gene, CYP2E1, and there are many other examples. This phenomenon has been described, has been called the metabolic zonation, and it's been studied with immunofluorescence, immunohistochemistry, or in situ hybridization, which are techniques that can reveal the spatial expression patterns of a few dozens of genes. And to understand how the liver works, we wanted to obtain a whole map, a map of the zonal expression of all of the hepatocyte genes. And this was a challenge until a few years ago. New technologies made this more feasible, and I'll tell you about one of the studies we've done in mice, and this was led by my amazing staff scientist, Keren Ba'ar-Alperin, together with a really talented computational student, Tom Shenhab. And they decided to use a technology that just emerged, which is single-cell RNA sequencing, where we take the liver and we dissociate it into thousands of cells. And for every cell, we're able to measure the expression levels, the RNA levels of all of the 20,000 genes encoded in the hepatocyte genome. So just to recap, all the cells in our body have the same DNA, but which includes 20,000 genes, encoding 20,000 potential proteins. But every cell type expresses a subset of these genes. So a skin cell would express a different subset than a neuron, and a hepatocyte would express a different subset than an intestinal cell. And if we can read out the RNA levels of all these 20,000 genes, we would know what is the function of the cell, what is the state of the cell. And so single-cell RNA sequencing for the first time enables us to measure the transcription, the expression profile of many hepatocytes. But the problem is that when we dissociated the liver, we completely lost the spatial identity of the hepatocyte. We didn't know if it originated from the central vein region or the portal vein region. And so to retrieve this spatial information, we utilized landmark genes, genes that we already mapped under the microscope, such as the red and the green genes you saw before. And so now when I look at this table that I have of the expression of 20,000 genes over 1,500 hepatocytes, I don't look at all of the genes. I only look at the genes I mapped under the microscope. And if I see an hepatocyte with 34 copies of the red gene and 2 copies of the blue gene, the red gene is a pericentral gene and the blue gene is a periportal gene, then I can infer that this hepatocyte originated from the central vein region. If I see another hepatocyte with 4 copies of the red gene and many copies of the blue gene, 50 copies of the blue gene, I can infer it originated from the periportal area. And this way I can build an algorithm that maps these 1,500 hepatocytes into layers. We used high resolution of 9 layers from the central vein outwards towards the portal vein. Confusingly, we used a different direction than the classical zone 1, 2, 3 because of technical reasons. And we were now able to group together all of the hepatocytes in each one of these layers, average their expression, and obtain the zonation pattern, the expression versus zone for all of the 20,000 genes of the mouse hepatocytes. And this is the map we got. So here, every column is a lobule layer, every row is a gene, and we have pericentral genes, we have periportal genes. Surprisingly, we also found the mid-lobular genes. So this is the classic zone 2, the mid-lobule. It was considered a transition area, but we found that some genes in mouse had specific expression in these mid-lobule zones. One of them is a very important liver gene called the HAMP, encoding the hepcidin hormone. This is a hormone secreted by hepatocytes that controls our systemic iron levels. And you can see here in the validation of single molecule fish that periportal hepatocytes have low RNA counts, the mid-lobular hepatocytes have very high levels of expression, and the pericentral hepatocytes have much lower expression. So this is a non-monotonic zonation profile. Well, now that we have a global map, we can ask whether specific functions of hepatocytes, out of these 500 functions, whether specific functions tend to be localized to specific zones. And indeed, we do see functional specialization. Periportal hepatocytes express much higher levels of expression of genes that encode for proteins that are secreted by the liver. These include albumin, that you can see here. Albumin is expressed in periportal hepatocytes about 3-fold higher than pericentral hepatocyte. This is albumin. But it also includes the clotting factors, the complement system proteins, any protein that is secreted from the hepatocyte. The hepatocyte is a factory for making circulating proteins. Making proteins is the most energetically expensive task of a cell. It takes up most of the ATP of a cell, and therefore it makes sense to allocate this function in the portal layers of the lobule, where hepatic artery brings in oxygenated blood. So there's more oxygen, there is more availability of ATP, and the hepatocytes can engage in energetically demanding tasks in these outer portal layers. In the pericentral layers, we see the specialization in liver detoxification, such as C2E1 here. But we could also ask whether there are enzymatic cascades that are not all performed in one zone, but are rather distributed across multiple zones. So for this, what all my students did is to parse the entire metabolic network of the cell. This is a really complicated mess of interactions. So every node here is an enzyme. An enzyme takes substrates and produce products that are substrates of other enzymes, etc. So this is the glycolysis, the TCA cycle. And basically what you try to do is to look for sequential enzymes that are expressed in sequential lobule layers. You try to see if there's sort of production line processes that are spread out across the sinusoid. And she found quite a few. I'll just tell you about some of them. So one of them is this following cascade. The enzyme glucose-6-phosphatase, which takes glucose-6-phosphate, removes the 6-phosphate, and produces glucose that can be thrown out of the hepatocyte. This is an enzyme that's expressed in the periportal hepatocytes, And the inverse enzyme, glucokinase, which takes the product glucose and phosphorylates it, giving glucose-6-phosphate, is expressed in the pericentral hepatocytes. So sort of the periportal hepatocytes feed the pericentral hepatocytes with the glucose. They produce glucose and that flows inwards and is taken up by the pericentral hepatocytes. Another very interesting enzymatic cascade that was distributed is this following enigmatic cascade of glutaminase 2, which is an enzyme that takes glutamine and produces glutamate. It's expressed in the portal layers of the lobule. And then there's the enzyme glutamine synthetase, encoded by this gene GLUL, that takes up glutamate and produces glutamine. And that's expressed only in the innermost layer. This is a very weird system, and it turns out to be related to one of the most important functions of the liver, which is ammonia detoxification. What you're seeing here is, again, this glutamine synthetase only expressed in the innermost layer, and ASS1, which is the key enzyme in a process called the urea cycle, which detoxifies ammonia into urea. Our body breaks up proteins all the time, and a toxic byproduct of this protein turnover is ammonia. And the liver is responsible for taking this ammonia, running it through a biochemical cycle, the urea cycle, and producing urea that is secreted in the urine. But in order for this cycle to work, the hepatocyte needs to also take up glutamine and produce glutamate. Now, if all of the hepatocyte would perform the urea cycle, we would get very efficient ammonia detoxification, but we would have a problem because blood would come in at the portal node with certain levels of glutamine and glutamate. And as it flows inwards and urea cycle is basically functioning, glutamine levels would go down, glutamate levels will go up, and then the blood would exit the central vein, come back from the portal node, and then glutamine levels again would go even lower, glutamate would go higher, and we would have an imbalance where glutamine would go to 0 and glutamate will go to saturation. So to prevent that, evolution is put in this inner layer that expresses glutamine synthetase that takes up all the excess glutamate that's a byproduct of the urea cycle that flows in the sinusoids and converts it back into glutamine so that the concentrations of glutamine and glutamate are equal in the entry to the lobule and the exit. Whereas in the middle, we had ammonia detoxification. Finally, another interesting production line process we found in our maps are related to bile acid biosynthesis. The liver takes up cholesterol, and from cholesterol, it produces bile acids. It takes up cholesterol from the blood, and produces bile acids that is thrown into the bile duct that flows backwards into the portal region and then into the gallbladder and the intestine, and is used to absorb lipids in the gut. And what we found is that the enzymes responsible for taking up cholesterol and producing bile were not all expressed in these inner layers, inner pericentral layers. Rather, we saw an expression in the innermost layer of the two first enzymes. And then in the second layer, we saw an expression of the third enzyme. And consequently, further enzymes were expressed even more downstream here. Downstream means towards the periportal region, because again, production of bile starts in the pericentral zone and flows through the bile duct towards the periportal region. So just to show you with the fish that this is really what happens, this is this enigmatic third enzyme that is actually silenced in the innermost layer and is upregulated in the second layer. And this could indicate that perhaps the process of bile acid biosynthesis is not performed in the same hepatocyte, but rather in several sequential hepatocytes, and that products are released and transferred from one hepatocyte to the next. So just to summarize this part, I've told you about a global map, a high-resolution map of hepatocyte zonation in mice that was performed using single-cell RNA sequencing combined with landmark genes. We found some design principles, some principles we think that confer optimality to the liver, principles of this division of labor. For example, allocation of highly energetically demanding tasks, such as protein production, also glucose production, gluconeogenesis, to these outermost layers that are highly oxygenated. We found production line processes where sequential enzymes were expressed in sequential lobule layers. We found enzymes that recycle material between different zones. There's other design principles related to zonal distribution of transporters with the different affinities and capacities that match the concentrations of the absorbed substrate. And this is all described in this review that Shani Ben Moshe wrote a few years ago. Okay, Natalie really beautifully described the implications of liver zonation. I want to really briefly repeat that. We think that studying liver zonation is critical, not only for understanding normal function, but also understanding pathological situations, such as regeneration after injury or different liver diseases. So just to give you two short examples. One of them is the acetaminophen intoxication. So in this beautiful plot here of the green hepatocytes and the red hepatocyte, the red gene is not just a gene, this is CYP2E1. It's a major enzyme involved in xenobiotic metabolism, taking up foreign substances, such as acetaminophen, called APAP, and converting it into a non-toxic substrate that can be thrown out of the body. And during this process of detoxification by CYP2E1, a very toxic molecule is created called NAPKI, and only the enzyme, only the cells that express the enzyme, only the pericentral hepatocytes would incur damage, because only they produce this NAPKI. Sorry, second. So nighttime saving in Israel. Okay, so you can see that only the pericentral hepatocytes are damaged because only they express CYP2E1. And then remarkably, this is 2 days after injection into mice of the equivalent of 1 bottle of Tylenol. And then 1 day later, you can see that the periportal hepatocytes go into action, start proliferating like crazy, and generate these new pericentral hepatocytes. And after 4 days, we have complete regeneration. And so this is, again, an important design principle, as we saw in the urea cycle. If all of the hepatocytes would engage in xenobiotic metabolism, after damage, we would not have these reserve hepatocytes that can go into action and regenerate the lost hepatocytes. And this is one feature that we think is important. Another maybe surprising pathology of the liver is the malaria liver stage. Malaria is a blood-borne disease of the parasite Plasmodium that basically enters erythrocytes, divides a few times, and kills the erythrocytes. But at the very first stage of the parasite, it has a compulsory liver stage. So the mosquito bites, transfers the parasite into the bloodstream. The parasite enters an hepatocyte, builds a vacuole, divides and matures, and then it kills the hepatocyte and ruptures it and goes and becomes the blood stage. And what we found together with Maria Mota in this work by my talented student Amichai Friat is that the Plasmodium parasite, depending on which zone it enters, has a very, very different fate. You can see here that if the Plasmodium infected the pericentral hepatocytes, This is a pericentral hepatocyte. The blue is the nuclei of the hepatocyte. The green dots are the RNA molecules of glutamine synthetase. And here, this green bag is the vacuole that contains hundreds of copies of the Plasmodium parasite. So this is a successful invasion, and this parasite in a day or two will rupture this hepatocyte and continue to the disease stage. This is if you end up in the pericentral region. If you end up in the periportal region, you often end up like this parasite. The vacuole is complete. The entire hepatocyte is green, and it is covered, is surrounded by immune cells that are going to eat it up. Okay, and this different trajectory of successful rapid development in infected pericentral hepatocyte versus failed abortive infections of periportal hepatocytes is related to the different biochemical composition of these hepatocytes. Mainly, there are many more goodies for the parasite around the central vein, and there are many more immune cells around the portal vein. Okay, so we'll stop with the mouse liver and move to the human liver. So all I've shown you was work in mice, and we're really interested in understanding whether these zonation features are relevant to human. And we want to describe the zonation map of human hepatocytes. And this is a project that we started about 6 years ago when a very talented surgeon joined my lab for his PhD, Orania Kubowski. And we immediately realized that reconstructing zonation maps of the human liver is much more challenging than mice. When we analyze mice, so the first question is, what is the healthy human liver that we can use as a reference? In mice, we always use 2-month-old male mice. They're all basically from the same family. They're all thin and very uniform. And when we think about human beings, we have different ages, sex, weight, associated pathologies. 1 in 3 healthy individuals has fatty liver disease. Okay, so it's not really a disease at the very earliest stages, but 1 out of 3 adult people has a steatotic liver. And so the question of how do we choose a healthy human reference was something that we thought about a lot. What we usually do when we analyze the healthy human liver is either use cadavers, liver tissue from cadavers, and these are very problematic to study gene expression because of the extended period of the hypoxia that changes gene expression in the liver. The second option is what we call adjacent normal liver. So basically, we use human liver lobes that are resected due to the presence of the tumor or a metastasis. And then we go several centimeters away from the tumor and take a piece of liver tissue. But again, the liver is the metabolic hub of our body, and it can be rewired and changed by tumors inside it. Even if it's not the tumor area, gene expression in this region could be changed. Actually, there's new studies that show that even a tumor outside of the liver, for example, pancreatic tumor or breast tumors, can rewire and change the gene expression of hepatocytes. And so the alternative approach we thought of was to use livers from live organ donors. The liver has this amazing regenerative capacity. Within a few weeks, you can remove half or 2/3 of the liver, and within a few weeks, it will grow to the same size. And we teamed up with Timuchin Tanner from the Mayo Clinic, who does a lot of these live organ donor transplantations. And together with Idon Nachmani and Itenkovich from Shiva Medical Center, who are doing this adjacent normal liver resections, we assembled a cohort of 16 patients, 8 live healthy donors, and 8 adjacent adjacent normal livers. And you can immediately see in this metadata that the live healthy donors are much more uniform in age, BMI. None of them are smoking. None of them receive chemotherapy. Chemotherapy can also change gene expression in the liver. This is a form of xenobiotic. And all of them have undergone the same surgery, whereas the adjacent normal livers are very variable in their age, their BMI, smoking status, associated drugs, associated diseases, and type of surgery. So for each one of these livers, we performed a range of what we call spatial transcriptomic approaches. And I will briefly tell you about this. So I told you in the mouse zonation reconstruction about one approach to reconstruct spatial expression maps, which is to use single-cell RNA sequencing of a dissociated tissue and to combine information from staining of landmark genes. In the last few years, there's been a dramatic development, biotechnological development that enables us to read out the expression of all of the 20,000 genes in intact tissue sections. This technique is called spatial transcriptomics. One of the technologies we're using for that is called Visium. I'll show you a brief video. We basically have a slide with the barcode, with the spots of 50 microns, where in every spot we have barcoded DNA molecules. Every DNA molecule in every spot has a different barcode sequence. And these DNA molecules can capture RNA. Now we take our tissue, we section our tissue on these slides, we stain with H&E and we take an image, and then we permeabilize the tissue. And the RNA just falls from the cells and is captured with these DNA probes that reside on the slides. Now we can amplify the probes, we can wash them off, and we can sequence them, and we can read the gene according to the sequence in every spot, how many copies we have of every gene, but also we know the location because of the barcode. And so in addition to H&E image, we get 20,000 images. This is a 50 micron resolution, but in a few slides I'll show you that now the technology also reaches 2 micron resolution. So this is basically a liver, a healthy human liver from a live organ donor. And this is these are the spots colored according to the expression signatures. So how do we reconstruct zonation? Again, we use CYP2E1. It's a classic central gene. You can see its expression here around the central vein. And we can use it as a handle to assign every spot one of 8 lobule layers. Again, we're numbering from the central vein towards the portal layers. And we did this for all the 16 cohorts. And then we put together all the spots from every layer, average their expression. And as in mice, we now obtain the zonation map of the healthy human liver. Every column here is a lobule layer, pericentral hepatocytes, periportal hepatocytes. And we can see that as in the mouse liver, more than half of the genes, of the hepatic genes, are significantly zonated and are changing. But now a very interesting question is, how does this zonation compare to zonation in mice? And the answer is that there are many, many discordances. So what you see here on the plot on the left, every dot is a gene, and the x-axis is the zonal bias, is the portal bias in mice. So the more to the right you are, the more portal a gene is. And the y-axis is the portal bias in human. And we thought we would see a very strong correlation, but as you can see, the correlation is quite low. So we do have genes like HAL that is periportal in both mouse and human. Mouse is red, human is blue. And we have pericentral genes that are pericentral in both mouse and human, such as CYP22. But then we have a lot of discordances. For example, this hepcidin gene, HAMP, that we saw as mid-lobular in mouse, it's actually portal in human. There are some genes that are pericentral in mouse and periportal in human. And most importantly, there are many, many genes that are periportal in mice and pericentral in human. And I'll just describe some of them because they're very important functionally. One of them is a gene called SLAC2A2, which encodes GLAT2. This is the main glucose transporter in the liver. Remember, one of the main tasks of the liver is to be the glucostat of our body. After we eat, most of the excess glucose from the meal goes into the hepatocytes. In mice, the expression of this transporter is periportal, and in human, it's pericentral. You can see the validation here. This is mouse liver. Green is the levels of the protein GLUT2, the transporter of glucose in mice. Red is the pericentral glutamine synthetase. So you can see in mice, the transporter of glucose is periportal, and in human, it's very strongly pericentral. And so in human, pericentral hepatocytes engaged in glucose absorption more than periportal hepatocyte. And this can explain a very interesting enigmatic feature of the liver that's been called the glucose paradox, namely that periportal hepatocyte, after a meal, generate glycogen not directly from glucose, but rather from lactate, whereas pericentral hepatocytes might generate them directly from glucose. Another important discordance between mice and human were immune-related genes. So Kupffer cell genes are periportal in mice, but pericentral in human. So the pericentral zone in human is more immune active. And finally, very important discordances, fatty acid synthase and lipid biosynthesis genes, they tend to be actually periportal in mouse and pericentral in human. So we have a map, we have a functional division of labor in human, and we see that there are large discordances that we think are important because when we want to model diseases in mice, we have to take these discordances into account. Our atlas also enabled us to study the earliest stages of fatty liver disease, which is the stage before inflammation, before steatohepatitis. This simple steatosis, accumulation of lipids in the liver, as Natalie beautifully showed you. Live healthy donors can donate their livers with up to 20% lipid content. So this is one of our 8 live healthy donors. And indeed, you can see that the lipid accumulation is pericentral. The pericentral hepatocytes accumulate more lipids compared to the portal hepatocytes. This could be related to this pericentral expression of fatty acids in TAS. Our atlas enabled us to use AI to classify the spots according to their lipid content. So for example, here we have a pericentral spot with a lot of lipids versus a pericentral spot with low levels of lipids. And now we could order our pericentral hepatocytes according to the lipid content and try to see if there are genes that go down in expression or up in expression as the hepatocytes become more and more steatotic. Okay, so this is the map you can see here. These are only pericentral hepatocytes. Every column is a lipid content. This is hepatocytes with no lipid content, and on the right are high lipid content. And there are many genes that change. We had 4 steatotic patients in our 16 patient cohort, and I'm just highlighting genes that are consistently changing in all of the patients. What we saw is that there is some decline in genes associated with a buildup of lipid droplets, fatty acid biosynthesis, like HSD17B13, and we see an increase in mitochondrially encoded transcripts. Now, this could be cool. This could indicate that as the hepatocytes become more and more steatotic, they change their gene expression to try to reduce the level of steatosis. For example, by increasing mitochondrial content, burning more fat, or by decreasing the levels of enzymes that build up these lipid droplets. But on the other hand, very enigmatically, we found that while the mitochondrially encoded transcript increased with lipid content, the levels of RNA of enzymes that work in the mitochondria but are encoded in the nucleus tend to decrease. So there is some discordance. These are genes that are usually stoichiometric, and in these early stages of steatosis, we see this breakup of stoichiometry, which could be also related to the eventual pathology. Finally, I want to tell you about the last feature of our human zonation atlas, and this is, of course, the other 5 cell types that Wynn described. The hepatocytes are not the only cell types in the liver. There are other cell types that are very, very important. For example, Kupffer cells that reside within the sinusoids that eat up bacteria that may have infiltrated, or old red blood cells, or even dying hepatocytes. There are the endothelial cells that line the sinusoids and the major vessels, like the portal vein, hepatic artery, and central vein. There are hepatic stellate cells, very enigmatic cells that can become myofibroblasts and start secreting collagen. And all of these cells are static. They reside within the sinusoid or between the sinusoid and the hepatocyte, but they're static and they sense the same levels of gradients of oxygen, nutrients, and hormones. And we wanted to ask whether they also exhibit zonation signatures, whether they're also changing their gene expression signature depending on their location, and how these zonated expression signatures could be related to interaction with zonal hepatocytes. So the problem is, to achieve this, we cannot study zonation of these supporting liver cells with our Visium spots because they're too big and they include many cells. Luckily for us, 10x Genomics developed an even higher resolution spatial transcriptomic technique called Visium HD, high definition Visium, which enables 2 micron resolution. And we took 3 of our live healthy donors and we applied this Visium HD. And we get these gene expression maps. You can see an example here of some genes, glutamate synthetase, which is around the central vein, SDS around the portal vein. And now what we can do is we can use the H&E to automatically segment individual cells, classify them according to size and morphology into hepatocytes in yellow, and the non-parenchymal cells, supporting cells in blue. And we can assign our 2 micron spots to individual cells and sum up their expression. And this actually enables us to get from the slide a single cell expression atlas, which we usually visualize with these U maps. So this is not a spatial map. Here, every dot is a segmented cell, and closed dots are similar in their gene expression signature. So you can see that we can clearly separate the hepatocytes from the non-hepatocytes. And according to gene expression, we can identify liver, sinusoidal endothelial cells, Kupffer cells, fibroblasts. But because all of this is from a single slide, we also record the zonation, the zone of every individual Kupffer cell or fibroblast or endothelial cell. This is the map here on the right, colored according to zone. Yellow are periportal cells, blue are pericentral cells. And now we can group together these non-parenchymal cells and obtain their zonation profile, which is what we did. And we specifically focused on trying to identify zonated ligands and their matching zonated receptors. And we studied all of the pairs of interacting cell types among endothelial cells, fibroblasts, hepatocytes, and Kupffer cells. And we found some very interesting interactions. For example, Wnt signals, such as this Wnt2 morphogen and responding 3, expressed by endothelial cells and fibroblasts, have their matching receptors, frizzled receptors, in hepatocytes or fibroblasts. And all of these, both the ligands and receptors, are strongly biased towards the pericentral zone. In the portal zone, we see expression of delta ligands, delta-like 4 ligands, and notch receptors between different cell types. Sorry again. And interestingly, we also found an enigmatic signaling from BMP molecules. So Wnt is known to be the master regulator of liver zonation. Recent years have suggested that BMP could also be involved. In our human liver samples, we have very strong zonated expression of BMP 10 and 9. GDF2 is BMP 9 by fibroblasts, and they're matching BMP receptors in hepatocytes and endothelial cells. And one hypothesis is that this could be upstream of the Wnt secretion of endothelial cells. So BMP and Wnt might be organizing liver zonation in the human liver. So just to wrap up, I've shown you a spatially resolved zonation atlas of the mouse and the healthy human liver. Healthy human liver presents distinct transcriptomic features and zonation features from mice livers. Early statosis is associated with gene expression changes and specifically some mitochondrial imbalances, an increase in mitochondrial RNA and a decline in nuclear RNA encoding mitochondrial enzymes. And that non-parenchymal cells have very strong zonation signature, including zonal expression of morphogens that could be shaping the zonation of hepatocytes. Finally, if you're interested in liver zonation and your favorite hepatic gene, check out our websites. We have some zonation browsers, such as this one, and you can enter your favorite gene of interest and examine its expression in the Visium HD, change with lipid content, see some examples of Visium HD visualizations of the lobules, and also expression in non-parenchymal cells. And I just want to wrap up by thanking many people: Oran Yakubovsky, the amazing MD PhD student who did the human liver study, Keren Bar Alperin, my staff scientist, collaborators at the Shiva Medical Center, Ido Nachmani Penkovic, the Mayo Clinic, Timuchin Tanner, Ido Amit Lab collaborated with us on the mouse study, and Maria Mota on the malaria. And I want to thank all of you for listening.
All right. Well, I want to thank both of you for introducing us to a brave new world. We have many questions, and so we'll go through some of them. The biggest question, which applies to both of you, I guess, is what are your thoughts about what really sets up zonation beyond oxygen and perhaps Wnt? What do you think of what are the elements that determine zonation beyond those? Natalie, what are your thoughts?
Yeah. So we saw that nutrient availability clearly shifts the function and the zonation. Shalev showed that at the RNA level, gene expression level, right? That feeding and fasting, and there is an element to clock genes that regulate that. I think there was a study that showed glucagon to counterpull against Wnt ligand in terms of gene expression. And I think there are other factors that are not unknown, right, and are yet to be discovered. Like, shall I just mention this BMP? Right, there are so many. This is an uncanonical, non-traditional compartment that for years was essentially invisible to us because we couldn't take it out. And yeah, I think there's still a lot to discover here.
So, Shalif, do you want to comment about that question?
Yeah, definitely. I mean, it's very complex, and there's a lot of graded microenvironment. There's one thing that we've been thinking a lot in recent years is whether hepatocytes, portal and central hepatocytes, are essentially the same cell type that just respond to different microenvironmental stimuli, or whether they're completely different, whether they're different as an hepatocyte and a neuron. So what really defines a cell type is epigenetic changes. So if I pluck out the pericentral periportal hepatocyte and I place them in the same microenvironment, same wind, same oxygen, would I get the same expression level of all the genes? Or will I still get regions in the genomes that are just unattainable in periportal and pericentral hepatocyte? So I think this is still open to debate. I can tell you that we had done some DNA methylation studies, and we did see differences. So DNA methylation is sort of an epigenetic mark that changes on a slower timescale. Pericentral periportal hepatocytes have some epigenetic changes. Another interesting feature that I think we saw in regeneration is that when acetaminophen causes the ablation of all the pericentral hepatocytes, periportal hepatocytes start to divide and bring about new pericentral hepatocytes. So basically, you get a pericentral hepatocyte. The 2 days ago was a periportal hepatocyte, and it completely changes its gene expression and becomes pericentral. But the interesting thing is that it doesn't directly transdifferentiate. It doesn't shut down periportal programs and upregulate pericentral program. What it actually does, it goes through a transient state of dedifferentiation and then redifferentiation. So it upregulates alpha fetoprotein and all sorts of genes that are only expressed in the fetal liver, and then it upregulates the pericentral genes. And that to us indicated that there could be more happening here. Maybe what you really need to do in order to become pericentral from a periportal hepatocyte is to actually completely rewire your epigenetic state. And I think this is a very interesting direction for the future, to study the dynamics and the zonal differences. And there are some nice, Stein Arts has a very nice work on zonation using a toxic, which is sort of an epigenetic mark. And this is, I think, a very interesting future direction.
There's a interesting question about the flexibility of zonation. Sort of, do we have any firm evidence other than some that was presented here as to how quickly zonation can change under different metabolic or environmental conditions or developmental ones? Is this a rather rapid dynamic process or something that's far more gradual? Natalie, you want to go first?
Yeah, sure. So this is exactly the type of question that my lab is interested in, the metabolic flexibility, what that is. And the liver, we know, needs to very rapidly respond to nutrients, right? When after an extended period of fasting, when food finally comes in, the liver needs to shut down gluconeogenesis, right? Otherwise, hyperglycemia takes place. So everything needs to be sensed and adjusted. And I think that is encoded by the organelles and mitochondrial dynamics and the ability of organelle to interact with each other and switch interactions, because those interactions are going to dictate where glucose is going, where lipids are going, and what's going to be their fate metabolically. So I think zonation moves, but because it's not restricted by membranes, the compartment is smaller. is subcellular, is at the organelle level, and that's where the switching takes place. And the gradients is constantly sensed. And, those changes occur depending on where the cell is in the tissue. I hope I answered the question.
Yes. Shalev, what are your thoughts?
Yeah, I agree. I think definitely the liver is highly responsive to glucose, of course, and other metabolites. Also, circadian rhythms, about 50% of the genes in the liver are zonated, and about 30% change throughout the day. I have to say, we did do a spatial reconstruction around the clock in mice. We did find that in terms of most of the hepatic genes, when they change zonation, when they change their level of expression, their zonation profile remains relatively the same. So that's the majority. I mean, if you have an increase or a decrease, sort of the zonation profile usually goes up and down rather than switching. But there are some very important exceptions, like Nathalie said, for example, gluconeogenesis. Gluconeogenesis is very portal in a fed state, and in a fat state, it becomes non-zonated. So there are also these examples. So definitely it's dynamic. I think the non-dynamic aspects are probably controlled by wind. But then weirdly, we found that also wind levels circulate around the clock. So that could basically control the relative height of pericentral or periportal zonation profiles. So some of it is static, some is dynamic, some change in amplitude, and some change in shape. Yeah, basically.
So, given that we all use models, whether they be animals or cells, but with the extraordinary differences, Shalev, that you've shown in studying of the human tissue, how cautious should we be when interpreting mouse models?
I think we have to be we have to understand the differences, right? So there's a lot of commonalities. Wind is a global organizer, and mouse work. There is no replacement of mouse experiments. It really allows you to do these beautiful experiments that Nathalie showed you. You can time perturbation. You can use genetic manipulations. So this is really the model organism to study something really mechanistically. But we do have to be aware of the. So for me, one of the most enigmatic features is the pericentral steatosis. So I mean, beta oxidation is periportal in both mouse and human, but fatty acid biosynthesis is actually more periportal in mouse, but pericentral in human. But both of them tend to accumulate lipids around the central vein, and maybe we can sort of hypothesize that accumulating lipid, it's basically an imbalance between you either you synthesize too much lipids or you don't burn enough. So maybe what's really important is the portal zonation of beta oxidation. This is just a region where you don't burn enough lipids. So maybe the synthesis is not that important, because both in mouse and human, you see this feature of pericentral steatosis. I do think that some, for us, like the differences in the hepatocyte and mouse zonation profiles was striking, and we're rethinking a lot about some of the hypotheses that we had. For example, immune zonation, what I showed you about malaria. This was a plasmodium of malaria, and one of the reasons that periportal infection failed is because there are more immune cells in the periportal regions in mouse, but in human there are more pericentral immune cells. So we don't know how it will behave in human, but of course it's almost impossible to study a disease like malaria in human. So yeah, so it's a trade-off. We just I think it's good to be aware of the differences, and definitely not to stop working on the mice.
So I'm curious, have the two of you, or Shalev, have you begun to examine mitochondrial dynamics in the human liver? Are you collaborating, or what can we look forward to?
Nathalie,
any student volunteers for intravital vivo imaging? Yeah, no, so I think that's definitely an interesting concept. In our mitochondria zonation, we do have some labeling of human samples, and we did look at mitochondria, lipid droplet contact sites also in the context of increasing steatosis levels in human patients. So it's possible, it's harder, and obviously you can't do the dynamics because the samples are fixed. So that's a little challenging.
But in this country, as well as in Israel, we have living donors, and we have extensive hepatectomies being done for people with cancer. Is there any collaboration afoot with more local people where you can do some of these studies dynamically, or is that in the cards?
So are you imagining like perfusing the human liver and kind of sustaining it alive so you can do a dynamic experiment? When is that where you're going?
Well, there's an interesting question. Pathologists have pointed out for years that in hepatic nodules, such as occur in cirrhosis, for example, the blood supply is about 100% arterial. Do those nodules show zonation? Do we know?
Yeah, I don't know.
I don't know. I think it's fascinating. There's some very interesting work from Neil Anderson, who studied some of these nodules. I think he focused on the non-parenchymal cells that surround the nodules. I don't remember if, I know this is something that comes up in the lab, and actually now that you're asking, we should probably do it if no one has done it. I don't remember if this has been done, but I think it's, the nice thing about this spatial transcriptomics is that it really makes studies much more feasible, right? You can go for archival tissue, paraffin-embedded block, section it, and the Visium HD works on fixed paraffin-embedded FFPE tissue. So you can go to any pathology, even rare pathologies, interesting cases, and simply read out with high resolution the spatial expression. So yeah, cirrhotic nodules could be really interesting, I think.
None of that's relevant, really. Let's see, we had another. Yes, so we have an interesting question that hasn't been touched upon, but do you think there's any relationship between the fact that these hepatocytes have the endothelial cells are fenestrated, and there was pretty extensive quantification of the relative size of fenestra, and there's really a gradient in fenestral size from the periportal to the pericentral. Does that enter into the equation at all, do you think, of what's going on within the cell? Have you ever?
Yeah, so Shalev, you did a study a few years ago with phosphorylation where you showed tyrosine phosphorylation is pericentral because of some endothelial marker or endothelial factor.
Yeah, this is together with Helmut Augustin, we did phosphoproteomics. We also did transcriptomics. There are many changes in gene expression in endothelial cells. So endothelial cells show profound zonation, including many interesting programs related to Notch, Delta, Ephrin. Endothelial cells around the central vein are very important. They produce angiocrine signals like Wnt. If you're asking about morphology, this is super interesting. I didn't know that the fenestration sizes or densities changed in a zonal manner. This is super interesting.
Can you remind me, are the fenestra larger in central regions?
Yes. No, in the periphery.
In the portal.
In the portal region.
That's fascinating, right? Yeah. In terms of modeling modeling the response to an individual. So sort of not just modeling the concentration. So the way we think about it is we have a concentration. As the substrate flows inwards, it's absorbed. So there's going to be like a linear decline in the concentration, but maybe it's like an exponential decline because it's that's really interesting.
Yeah, and
I didn't know
there's you need to add to that model also the expression levels on the surface of the cells, right, of the receptor or whatever. Which, by the way, reminds me, you showed the distribution of the glucose transporters and the difference between the human and mouse. But what was interesting, you showed that the human have more periportal on the surface, and in mice it's pericentral, right?
No,
Or was it
the opposite.
the other way around?
Yeah.
In any event, in the human, you have a huge intracellular pool that is not on the plasma membrane, kind of ready to go. So in the membrane trafficking world, and that stands out when you look at those two images side by side, you see that in the mouse, a lot of the transporter is on the plasma membrane, maybe not just in the hepatocytes, but also endothelial cells. But then in the human tissue, it's very clear that there is a large pool that is cytosolic. You have a green signal in the cytosol that perhaps is ready to go for when the time is right.
Yeah, like GLAT4, basically like GLAT4.
Right.
Interesting. Yeah, of course, localization could play, of course, an effect in the absorption characteristics. Yeah, interesting.
So here's a very important question that we've received. When cancer hijacks zonation, which the study that you presented, I think from Texas, suggests, does it copy a specific zone, such as a zone with having oxidative stress? Does it involve mixing zones, or does it really break the rules entirely and have nothing to do with those substrates? I guess it's a little early in terms of information, but what are your thoughts about that, Natalie?
Yeah, so I mean, what they did in that study is pretty remarkable. So they developed these mice lines, pre-lox mice line that allows them essentially to genetically engineer the tumors in specific zones. And they introduced these two commonly mutated genes, and those are common in human HCC. They introduced them either in periportal or pericentral. And what they show, again, it was very surprising, while the periportal tumors are growing and expanding, it's HCC only develops in pericentral zone 3 cells. And the question is, they focused on what gives advantage, or essentially the healthy zonation physiology that is protecting the cancer cells and essentially promoting tumor progression in the pericentral. I think an interesting question could be what protects periportal hepatocytes from this same transformation, right? They introduce the same oncogenic drivers in two different environments, which makes a very strong case, at least in liver cancer, that location matters, right? And if you have cellular transformation in periportal, it's not as bad as if it occurred in pericentral. And in fact, there are some sequencing studies in humans that showed that patients with periportal signature tumors have better outcomes with those of pericentral signatures, right? But I think this is the first study. Well, I don't want to say the first, because there was one study before that, a PNAS study, where they gave the mice a chemical in the drinking water, and they did cell tracing, and they also showed that the tumors HCC develop primarily in pericentral regions in another model. But I think this is really the first time where there is a genetic control of the location of the cellular transformation, and very striking and interesting evidence to support that, one, cancer is a spatial disease. Zonation matters for cancer, for diagnostics, diagnosis and maybe even for treatment. And the other thing is, why? What is it in the environment that is permitting tumors to develop in one side of the lobule, not other? Is it like what Shalev mentioned earlier with the malaria? Is it the immune presence? Our periportal regions, at least in mouse, are populated with more immune cells that could serve as a defense line and recognize tumors or transformation, or is it something else?
Okay. So, Shalev, we have a malaria question. Apparently, the intracellular vacuole that you described, in which replication of the malarial spherocytes takes place prior to their egress, that's an acidified component for which I believe there's a fair amount of evidence as to what the composition, what's going on in that vacuole. So how do you interpret the findings that you showed, which are really quite dramatic, that apparently in the periportal area, the spherocytes can enter but not replicate? Or how do you interpret how do the malarial experts interpret the findings that you dramatically observed? And there's another part to it, because there is evidence for receptors on hepatocytes, and there's a big argument as to exactly what they are, but I wonder whether there's been consideration of the distribution of any of those candidate receptors for the plasmodium.
Yeah, thanks. Thanks for the question. So it's not that the image that I showed you was not the representative case. So there are successful infections also in portal hepatocytes. Statistically, they tend to develop more slowly and to have more abortive infections. The reason we think, what we are hypothesizing, some of the things that malaria parasites need, they need two things. They need hem, and they need lipids. They need lipids to build membranes, because they divide this syncytium. But then before they puncture the hepatocyte and are released, they need to build membranes and become hundreds or thousands of cells. And there's more lipids in pericentral hepatocytes, as we've seen, right? There's more lipid content, more fatty acid biosynthesis, or less burn down, beta oxidation. Hem is also an important molecule for parasites. There's a lot of hem in cytochrome P450 complex. So that could be one reason. Second reason could be really the immune infiltration and the sensitivities to infections. It could be that the periportal hepatocytes, because this is where the sites where you're also exposed to bacteria that might be infiltrating from the gut, you could raise a red flag with a higher sensitivity and attract immune cells. Now, in terms of the infection, although we saw that pericentral hepatocytes have infections that develop faster and more successfully, the initial infiltration is random. So this we showed. There's a very interesting feature of the parasite. It doesn't settle in the first hepatocyte. It enters an hepatocyte, then it drills within the hepatic wall, and on average, it visits 3 hepatocytes until it settles and builds a vacuole. And we also asked, whether this integration is random, whether it senses the host and decides on an optimal host. So we had, in addition to the green plasmodium, we also had dextrane injected to the mice, and that enters into the hepatocyte and stains every cell that was visited. And so we sorted the green hepatocytes, which are the one colonized, the yellow hepatocytes, which were the one that were visited but not colonized, and the non-labeled hepatocytes, which are all the rest. And we didn't find differences. So it seems random, but then the eventual course of progression is very different, depending on zone.
Of course, if it were possible to control the entry or the spherocyte formation essentially within the vacuole, you'd prevent the disease.
Yes, there is one interesting direction is that these abortive hepatocytes that I've shown you. By the way, there's a crosstalk between the host and the parasite. So as I told you, one of the things that, two things the host does to limit the plasmodium. One thing is lower fatty acid biosynthesis. Again, the plasmodium needs fatty acids. The host significantly lowers fatty acid biosynthesis expression. And it also upregulates iron buffers to deplete the iron from the parasite. Really, this is very striking, and we saw it both in fish. In terms of treatments, one very interesting, potential future therapeutic, these abortive infections where the vacuole bursts has a very, very unique hepatic signature. It has an upregulation of P53 and of Notch and MYC. All these pathways are dramatically upregulated. So in principle, if you could induce this cellular state, it could be maybe driving abortive infections. But again, this is very futuristic direction.
Very cool.
So, Shalev, we have another question apropos of the Tylenol studies. Folks remember that quite a few years ago, I think it was Jerry Mitchell at the NIH, who presented the evidence that reactive products due to the effect of reactive oxygen and so forth, and it was the really depletion of glutathione that gave rise to that activation, which then caused disease. So do we know about the zonal distribution of glutathione and its pathway?
Yeah, it's a good question. Of course, this is very important, and the N-acetylcysteine can also, lower these reactive oxygen species and improve outcome. I don't remember off the top of my head. We have, we have these Atlas browsers. I sort of vaguely remember it was enigmatic. It was not what we thought, but I don't remember. But we have, in our website, we have an atlas of all the time points after regeneration and the expression of any gene, in controls and in all the cell types. It's a very interesting question that I will check after the talk. Thank you.
Okay. A very important question we'd like to hear both of you comment about. And what do you think is next from the technological standpoint? With higher spatial resolution tools, what are the next big questions both of you are excited to investigate? Natalie, you want to go first?
Yeah, so we're very interested, as I mentioned, in the organelle remodeling, the membrane contact sites, and how that is used to regulate metabolic output. So if you think about just the mitochondria lipid droplet contact sites, and promoting these contacts lead to lipid storage and lipid droplet growth, and dissociating it makes the lipid droplet go away. Is that a good target to reverse fatty liver, essentially, right? As simple as that. What are the functional requirements? And, it's like switching gears. Which organelles need to touch which organelles to get the metabolic consequence that you want? And how can it help us reverse fatty liver and prevent cancer? And or treat cancer by shifting the cell's metabolic state, essentially. That's kind of where we're going conceptually. Technologically, I think we are pushing to move from the transcripts. I think the Shalev showcased very nicely how Visium and spatial transcriptomics is really at the peak. We need maps of proteins. So we just we're publishing, it's It's coming out on Friday. Collaboration with Matthias Mann, where we did single-cell proteomics in human samples. So creating maps at another level, right? The proteins, that's closer to function. Later on would be the metabolites, which is really the byproducts.
Okay, you don't have to give your whole plan for the next. Shelley, what is your view about it from your standpoint of you see as both the technological and the major issues you'd like to address?
Yeah, I'm really excited about the use of these new technologies to study paraffin-embedded tissues. You know, we have pathology banks with many, many, many tissues, and one thing we're very interested in is you know, other infections like hepatitis B virus, how that depends on zonation and progression. I think the whole process of muscle, the metabolically associated liver disease. So we sort of touched with a few individuals in the very earliest stages. There's very nice work on the later stages, steatohepatitis, but I think it's heterogeneous among patients. It's very interesting to follow the dynamics and really to study the mechanisms, because this is a really important disease, the 21st century. And now I think we have the availability to really tap into that. I think also it's very nice when you can study human tissues, you can think about also mechanistic experiments. There's one example I can give you is portal vein ligation. This is sort of an initial surgery that's done before you remove a lobe. Basically, you shut down the portal entry into the lobe you're going to remove, and then the other lobe works harder, so it grows. This is a lobe with a tumor. The other one grows, and then you have enough liver mass, so you can go then with another surgery and remove the affected lobe, because now the other lobe is bigger. But now we have the lobe that you have one lobe that had both arterial blood and venous blood, and another lobe that had only arterial blood and not venous blood. So if you can sample these two lobes, this is sort of an experiment that can study zonation in both of them and sort of discern the effect of oxygen concentrations versus blood-borne hormone and nutrient concentration. So this is sort of an experiment you can do in you know, from a surgery room in human. So we're sort of trying to think about these kind of direction on human livers, and yeah, we'll see where it goes.
I think it's very exciting that between the two of you, you know, sort of help to build part of a bridge that's connecting cellular biology with human disease. Both have much to contribute, particularly the human disease, because there are human diseases you can't reproduce, or at least they're not valuable in experimental animals. But you wonder about what would happen to the kind of measurements that you've made, Shelley, for example, in people who have prolonged cardiac failure with venous backflow without thrombosis, or even if somebody has an early stage, you know, sort of Budd-Chiari type of portal vein obstruction. And then there are many other examples where in the human disease spectrum, something dramatic must be going on, not just the metabolic changes, but actually in the vascular changes and in the regenerative changes. And the opportunity to do this, as you've shown in human liver, is really breathtaking and provides further clues for people who work primarily as with the systems that Natalie uses to explore more fully. So I really think this is a brave new world. Parenthetically, I would point out that it turns out in the kidney, the three regions of the proximal and convoluted distant tubule are in the same state that the liver zonation was roughly about 50 years ago. There's evidence of different enzymatic activity in different parts, but exactly what's going on and where, and particularly in renal disease that is associated with impaired tubular function. So this may be something of much broader implication, even beyond the liver. So on behalf of all the people listening and personally, we're all delighted, and I trust that everybody is excited as I am to hear you present your exciting work, and look forward to hearing more in the future. So thank you again for participating. I have to announce that the CME code is 67270. So thank you,
Thank you.
and go well.
Thank you.